I would like to thank the organizers of the meeting for this exceptional opportunity to present the work which we are doing in Berlin right now, which started a long time ago in Strasbourg. And I also want to stress the incredible conference, the scope of topics which are covered here. And during my life, I've never been to such a conference which focused specifically on insects and looked from all angles. That's really, really very nice. Thank you. So I will start with just several facts, and we forget often that 17% of all infectious diseases in humans are caused by insects which transmit the pathogens which induce these diseases. And malaria, one of the major killers among the infectious diseases transmitted by insects, is obligatory transmitted by Anopheles mosquitoes. So only Anopheles species can transmit human malaria. However, among these Anopheles species, which are about 300, 400, which are known right now around the world, only a tiny proportion, not more than 10%, really can transmit malaria and worldwide. So there is this bottleneck of the transmission where there is a very precise interactions between the vector and the parasite, which make the transmission possible. And this is really a very weak point of the transmission. So why do we still deal with this enormous burden of malaria in the world if these interactions are so delicate? And this is the question which we are asking in our research. What makes the malaria vector? Why mosquitoes which transmit malaria do so? What is necessary for the parasite during development in the mosquito? And first, I will briefly remind you the life cycle. I know this is the moment when everybody starts to look at their phones, but I think it's very important to understand what happens between the human side, which is on the left side of the screen. And usually when you read publications about malaria, the mostly discussed is this part of the cycle, which is in fact is a kind of a reservoir of the parasite, because the major event in the parasite life, the sexual cycle of the parasite, happens in the mosquito. So the gametes which are developed in the human, in the blood cells, can only transform into gametocytes inside the mosquito gut. So this is the first step which is important for the transmission, because if there is no formation of the gametes, then there is no transmission. This is the dead end of the parasite. They could cycle in the human and die inside the human blood. However, mosquito picks these gametocytes, and the development of the parasite continues. The parasites really very rapidly, within 10 minutes, fuse inside the mosquito gut, form a mobile form which is called oocanid, which will penetrate the first epithelia of the midgut of mosquitoes and establishes infection on the other side, where the parasites will multiply. After multiplication, which takes, depending on the species, between 1 and 2 weeks, the parasites will transform into another form, which is completely different from the form which was taken up by the parasite or migrated through the gut. And this form is called sporozoites, which will again migrate and invade salivary glands of the mosquitoes. So basically from the midgut, which is here, it will reach salivary glands in the thorax. And there, they'll become infectious for the next human bite. So if you look in the duration of the cycle, as I said, it's approximately from the infection time, which is 0 here on the slide, to the moment when mosquitoes become infectious, which is in the case of Plasmodium falciparum around 2 weeks after the infection. The parasites really change enormously in their shape, in their identity, and also what's interesting, they change in the numbers. Because from the number of gametes and gametocytes which are taken up by mosquitoes, which is in hundreds, in thousands, the number of the parasites which develop on the mosquito oocysts is really very little, between 10 and 100 oocysts per mosquito midgut. But then each of these oocysts will give rise to these hundreds of thousands of sporozoites, where there is the amplification of the parasitic life cycle, which mediates this transmission. As a biologist, of course, we are very attracted by this drop in the parasite numbers, during this first step of interaction between the parasite and the vector. What happens at this stage, and what determines this drop in the parasite numbers? Why not all oocytes will make it into oocysts? And that was an interesting question, which really was in the center of investigations by multiple groups. And in fact, it became clear that there are some genetic factors in the mosquitoes which really prevent development of oocytes in the midgut of the mosquitoes. And somehow by chance, but looking at the conserved immune molecules in the mosquito genome, that we came across the family of thiaester-containing proteins, and we called them back like that because they have a very important molecular signature, a thiaster motif, which is located in the domain, which makes them the close relatives of the complement system in mammals. Now, complement system is very potent innate immune response, which we all have. And the major characteristic of this response is that this molecule is capable of recognizing pathogens inside our body and bind covalently to the surface of these pathogens and either promote phagocytosis or induce direct killing of the pathogens by lysis. Now, mosquitoes also have the same system, it appears, and what they do, they use this to kill the parasites. We could really observe binding of TAP1, which is in red on this immunofluorescence image, to the surface of the invading oocytes in the midgut. And we could show that these parasites are dead. Now, of course, if you see these images, we still can wonder whether TAP1 binds to dead parasites or binding of TAP1 induces parasite killing. And the answer came from adaptation of the double-strand RNA injection, or RNAi, approaches in the mosquitoes, which was in the early 2000s, and we were very lucky that it works very well in our organism. And when we inhibited TAP1 by injecting double-strand RNA directly in the mosquitoes, we could see that in the absence of TAP1, the numbers of the parasites are dramatically increased. And that was very clear that those parasites which don't make it to the oocyst, in fact, are eliminated by recognition of TAP1 of the surface of the oocytes. So if TAP1 is not there, then the parasites are not eliminated and they can freely develop inside the mosquitoes. And that was really fully susceptible mosquitoes, which we consider to be really very highly susceptible to the parasite infections. Now, interestingly, so that was the first genetic factor which was identified in the mosquitoes, which kills parasites and prevents amplification of the parasites inside the mosquito. However, as you see on this slide, not all parasites are killed by TAP1, and some of them can evade this surveillance of TAP1 and develop further. And interestingly, there is TAP1 is a highly polymorphic gene. So I just plotted the schematic representation of different major allelic classes because we are not speaking about single SNPs here. If you look at the crystal structure of TAP1 in comparison between very divergent S1 form and R1 form, indicated here by different colors, you see the accumulation of these mutations or variations, variable amino acids on the structure, which are mostly concentrated on this working head of the molecule, the thiaster domain, and around the thiaster site. And there is very few of those, but still they are present in the stalk of the molecule which supports this head. So this is extraordinary polymorphism. It is one of the most polymorphic genes in the mosquito genome, and I think by the level of polymorphism, it can be only compared to the sodium channel, which is responsible for insecticide resistance and is under very huge selective pressure in natural populations. So what is interesting that polymorphism in TAP1, so if we look at different classes and do genetic crosses between mosquitoes containing different alleles of TAP1, what we could see that there is a progressive increase in resistance if we are having mosquitoes homozygous for this R1 allele, which was called R allele because of the resistance of the mosquitoes to the parasite. And other alleles here are two. Immediately there is a drop in resistance and appearance of higher parasite numbers, and S2, which is also even more susceptible allele of TAP1. So that was very interesting because it seems that there is a selection in the mosquito towards appearance of this resistance to parasites, which can be probably induced by interaction between mosquitoes and the parasite in natural situations. And this is summarized in the slide. They are type 1 in females because, unfortunately, we do not have a gender equilibrium in the system, and only females transmit malaria, so we mostly focus on analyzing females. Polymorphism in type 1, and specifically R1 allele, is important to protect mosquitoes from malaria infection. And when we looked at males, and sometimes you have to work with males, and it was very interesting because they taught us something yet interesting, because the same mechanism which mosquitoes use to defend themselves against malaria is also used in fertility check in the mosquitoes' testes. So type 1, we found type 1 which was bound to the defective sperm in the testes of mosquitoes. So here we could see that type 1 function is not only directed against pathogens and non-self, but can also recognize defeat self and clear it up for the normal functioning of the organism. And again, immunity and reproduction are probably the most strong drivers of selection in natural populations, because these functions are completely required for maintenance of the species. So now we had a situation that out of 4 allelic classes, we could identify functional relevance in resistance to the parasites and infertility, whereas 2 other alleles, S1 and R2, we could not define any phenotype. So this is where the work in the lab starts to be, comes to its end, because how can we create conditions in the lab and to understand what this polymorphism really brings to the species of Anopheles gambiae, how we could really read this importance of pleiotropic polymorphism of type 1. So to gain more insight into this polymorphism and its evolutionary significance, we went to Africa and we asked the question, where do we find these alleles in natural populations of mosquitoes? Because so far we were working with mosquito lines from the lab, which were in the captivity for at least 30 years. And so we visited several countries across Africa, like Mali, Burkina Faso, Cameroon, and Kenya, where together with the collaborators we collected mosquitoes and genotyped them for type 1 and for species. What we found that these alleles, which we know least about, are the most dominant alleles. They were found in all mosquito species in Africa and also in all countries. So those are the generalist alleles of type 1. They are the most prevalent alleles in the field. And as you remember from my first slide, in laboratory conditions, these alleles will not confer resistance to Plasmodium. In contrast, R1 allele was specifically found in sub-Saharan parts in Mali and Burkina Faso, where it is really very dry and arid climate. Interestingly, the S2 allele was specific to Central Africa. And in fact, we did not identify populations where we have mosquitoes which would have at the same time R1 allele and S2 alleles. These alleles are really separated in time, and basically what was discussed earlier, there is this speciation which leads to really specific development of particular alleles. So there is something in this geographical separation which drives selection of these alleles in the same mosquito species. Now, if we look at this genotype now of the mosquitoes, taking into account the individuals and double complement of the alleles, so what we could see, so this diagram represents collected mosquitoes. On the lower part of the diagram, the species which we identified in our collections. It's important also to mention that these species are morphologically identical. You can't say them apart if you just look at them. So there is no way to distinguish them morphologically. You can only do genetical analysis using some markers to define the species specifically. And inside the species, Anopheles merus, Arabiensis gambia, and Anopheles causality, you see the majority is Anopheles gambiae, sensu stricto. So it's different from Anopheles causality, although we, until very recently confused one with another. So we believe that it was all Anopheles gambiae. Now, Anopheles gambiae, the major class is susceptible S1S1 and S1R2 genotypes, whereas in Anopheles coluzzii, the major class is R1R1 homozygotes, those which will be resistant to malaria parasites, and some proportion of S1S1 and S1S2 and other minor combinations of the alleles. Now we ask the question, where do we find these genotypes in natural populations? And we mapped the origin of the collected mosquitoes according to these geographical and climate positions around the world. And the species are coded by this shape of the symbols. And what we could see very clearly that Anopheles coluzzii, which is a round shape here, very separates according to the humidity gradient and is present mostly in arid areas, as I said before, in very hot, semi-dry areas. Importantly, in the same color, we found also in the same position, in the same location, Anopheles gambiae, as this yellow triangle shows, but those don't separate into this gradient of aridity, and basically there is no separation in climate between Anopheles gambiae and Anopheles coluzzii from wet areas in Africa, suggesting that there is some component in climate which drives selection of these resistant alleles in natural populations. And with that, what we proposed, that there is a really allopatric speciation in this Anopheles coluzzii specifically. Anopheles coluzzii is only present in Western and Central Africa. It is totally absent from East Africa, where is only Anopheles gambiae present. And, but these are the areas of the major malaria outbreaks and endemicity, highly endemic areas in malaria transmission. And what we see that R1 allele is really localizes in this African transient zone, which is at the limit between Sahara and the savanna areas indicated here by green, and the more generally spread S1R2 forms are closely located to the more humid areas, and S2 is only enriched in the Central Africa. Well, that is very interesting, and it gives us some ideas about specific organization of vector populations in Africa, but does it matter? Do we have to care about this allelic composition and about complex species in various countries? Is it relevant for malaria transmission? Or any mosquitoes, as long as there are mosquitoes, they will transmit malaria and drive disease into human populations? And so to answer this question, we selected this particular site in Mali I mentioned before, where we found both Anopheles coluzzii, which has this resistant genotype R1R1, and Anopheles gambiae, which has the susceptible genotype S1S1 or S1R2. And what we did, we decided to collect samples and as a time series throughout the whole rainy season. So the student of the lab, Marcus Gildenhardt, had this idea and went to Africa with our collaborators to start collections. So we started collections in early May before the season started, and we put traps in the houses in the particular village where we found this sympatric localization of mosquitoes. We also put a weather station in this main central electricity house of the village, where usually villagers charge their phones. And we also collected larvae at different stages also throughout this rainy season. So just to give you an impression, but this is the same site, and the pictures are taken at the different periods of the rainy season. So it's really completely different environment if you look at the site somewhere in June, where there is only impermanent places where larvae can grow after rain, basically, and then going more closer to the August, where you start to have more permanent larval breeding sites. So you can orient yourself according to this dry tree, which is found on all pictures. And then to the peak of the season, somewhere in September, and then the stagnation of water, very rich in biological debris versus the end of the season. And so what we collected, mosquitoes, we trapped them, so it is really very unsupervised collection of the samples. And what we could see that there is a really dynamics of mosquitoes in this area over the transmission season. There are very low numbers of mosquitoes in the beginning, so basically corresponding to these two climatic conditions. And we see that Anopheles coluzzii, which is resistant genotype, is the predominant vector in this area. And Anopheles gambiae is present in the more modest numbers. And we also see that the number of infected mosquitoes shown in red is relatively low comparing to the total number of mosquitoes during the whole season. And it's about 10% of all mosquitoes which will have parasites inside. So we genotype total mosquitoes using the COX-1 gene of Plasmodium falciparum to identify mosquitoes which contain the parasites. And of course, now the question was, can we identify the factor which will drive appearance or abundance of these infected mosquitoes in the population? Would it mean it's a kind of a correlation, but correlations can be very tricky. And this is a very famous paper appeared in the New England Journal of Medicine, which basically suggested that chocolate consumption, cognitive function, and the number of Nobel laureates per country are basically connected. There is a direct correlation. So more people in the country eat chocolate, there is more in the country number of Nobel laureates. And it seems working very well for Switzerland. They higher consumption and higher number of Nobel laureates. It's not working very well for China at the moment. Maybe it will change. France is somewhere in the good place in the middle, and there is some problem with German chocolate, I think, because they eat a lot, but the number of Nobel laureates doesn't really augment proportionally. So correlation is not causation. We can say that if the country eats more chocolate, there will be more. So it does not drive the number of Nobel laureates per country. So we wanted to come up with a method which allow us to predict, to be causal to what we are studying, and we turned out to the econometric method, which is called the Granger causality. And I won't bother you with the details of this mathematical assembly. I just want you to understand the principle of the method. And the method is based on the comparison of building models of two variables which are taken in a time series. For example, we look at the coffee prices of a period of time. And we compare the coffee prices of Arabica, which will be our X variable, and the coffee prices for Robusta. And of course, it's a market, there is also variation in the prices, and so there is this nervous line which appears throughout this time span. But you could see, and this is on purpose very detectable, for example, this peak in the price of Arabica then later causes, or let's say, correlates with the increase in the price of Robusta, and that we see on several occasions throughout this time series, right? So the idea of the Granger causality test that to predict the signal X, for example, or of Y, it means that you need to know the time series, both time series, and not just single one. So you need the components of this time series X and Y to predict Y, and vice versa, right? So what we are doing, we are predicting. We are not correlating. We can't pick it up by eye. So we try to use mathematical approaches to predict what will cause causal and non-causal effects. So in our case, so the strength of the method, because it shows and it evaluates statistical significance of the model, how well model predicts one or another series, and it goes in both directions. It gives some statistical sign, and it also shows the direction. So is it a negative correlation or a positive correlation, like in this sense, if the prices are higher for Arabica, they will be higher for Robusta. Now, to our data, so what we were interested in is to predict Plasmodium abundance, so number of mosquitoes which will have parasites inside them. And for that, we prepared models for Anopheles abundance, so total mosquito abundance. So if there is more mosquitoes, will be increase and predict the number of infected mosquitoes. or we used only Anopheles causality abundance, or Anopheles gambiae abundance, or the ratio between Anopheles causality and Anopheles gambiae. Basically, this could go Anopheles causality prevalence. And we also took some weather parameters such as temperature and rainfall, which are very often used in modeling approach to predict in epidemiological models. And what I can say immediately that the weather parameters didn't work at all. In our modeling, there was really no causality for weather parameters, either temperature or rainfall. And the summary of our results is here. So if you use this method, what we've seen, the total mosquitoes also were not predictive of the number of infected mosquitoes. So it doesn't matter that there are more of infected mosquitoes to see of mosquitoes, total mosquitoes, that there will be infection. And on the other side, also number of infected mosquitoes did not predict the total number of mosquitoes, which is good. However, very importantly, the percentage of Anopheles causality mosquitoes, the prevalence of causality in the population, was really causative for the number of infected mosquitoes, and it was a negative interaction, meaning there are more of Anopheles causality mosquitoes, fewer will find infected mosquitoes in the population. And also importantly, and completely unexpected, the number of infected mosquitoes did not change the proportion of species in the population. So these results showed that population structure predicts, so if we know what is the prevalence of Anopheles causality and Anopheles gambiae, we can predict the number of infected mosquitoes in the populations, and also that Plasmodium infection, at least in this site, do not have any selective pressure on the mosquito genotypes in the populations. So what did we learn from this? Is that in very controlled, very clean laboratory experiments, we could show that vector competence is genetically determined, and identified mosquito complement-like system as the major factor which determines outcome of infections. In the field, in really very chaotic conditions, when it's always mixed with animals passing and drinking from the breeding sites and taking some larva collections from us, we could show that vector genetics drives Plasmodium abundance. And of course, which is important conclusions, that Plasmodium does not impose selective pressure on mosquito populations, and that the question remains, what factors actually drive mosquito microevolution in Africa? So why is it important to know? And of course, the first one, if we could identify factors which drive composition of mosquito populations, we can predict how that will change in the case of climate change, right? Which is a very important topic and will be addressed by Annabella later after me. And the second, very maybe more into kind of important application is the development of gene drives, and you know this drive to use those very fast in populations to also to curb malaria transmission. However, this application is very precise. It's really the top art of what we can do in genetic engineering right now, but it has to know what to drive, right, and which populations to drive, because it depends on the crosses between the mosquitoes to pass in this modified genetic information. And I must say that at the moment, we only culture in the labs Anopheles causality mosquitoes. There is no way to put into culture Anopheles gambiae. And as the results of our study show, this is the major transmitter. So if we will hurry up and release the first genetic drives which we can have at the moment in the labs, we basically will collapse populations of Anopheles causality, which, at least in Mali, are not the major driver of malaria, which will probably result in the failure of this method and which will be attributed to this inefficiency of genetic tools. And I think we have, as a scientist, responsibility to use precise tools in the precise locations. We have to be also precise in what we want to do, where we want to do it locally. And with that, I would like to thank people who contributed to the study, and it's Marcus Gildehart, very gifted PhD student in the lab, also Paolo Carilla Bustamante, who is a molecular modeler in the lab, and Evan Sronek, who contributed to this massive project of mosquito genotyping. And of course, it wouldn't be possible with our colleagues in the University of Bamako and also villages of Nankilabougou, where the samples were collected, and then also other colleagues for different institutions in Africa and worldwide. And thank you for your attention.
We have time for one question, please.
So, Elena, very interesting data, and I was curious that you showed this negative correlation of the percentage of coluzzii in the population, but you did not show if there was a correlation between the number of gambiae, that's in theory the sensitive case. So I was wondering, do you have a positive correlation with the population of gambiae? And if not, this suggests that it's a lot more complicated, so you need a certain proportion of sensitive and resistant. I mean, could you comment on that?
Yeah, as it is a prevalence, so it works a reverse way for gambiae. So I could have shown the results as proportion of gambiae, which will positively correlate with the infection. But at the level of abundance, we did not see it for both seasons. We've seen it only for season 2, where we have much more robust collections comparing to, like, it was 6,000 mosquitoes at that time. So I guess if you really intensify these collections, you could get to this abundance. I think percentage is much more sensitive and reveals.
But you assume then it's just a matter of abundance. So if you have enough of the sensitive, then you get transmission.
No, it's a matter of composition, what it says. So the composition is the most important. Then comes the number, of course, because composition is based on numbers. It's just percentage. It takes away this total number, right? It's regardless how many mosquitoes you have in total, which is also what we showed through this mathematical approach. But it's important the composition, the ratio between species. So if you would have more of gambiae, you will have higher levels of transmission if you have the higher number of causality.
Thanks very much. I think we should move on to the next speaker. Thanks again.